From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 60, recorded on May 26, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hey, Vincent. Hey, Tim.
And from New York, Tim Chung.
Hello. Hi, everyone. Hi, Vincent. Hi, Jason. Good to be back.
We were expecting Vivian, but she's going to be late, if at all, so we will forward on without her. If you enjoy these science programs from Microbe TV, we have like 9 different podcasts in different areas, and we talk about science, real science, no lies, no misinformation, and we need that these days. If you enjoy these, please contribute and help support us. We don't have advertising in our podcasts. We depend on listener donations. And, it's not free to make podcasts. Once you get to a certain point, we have editors working for us because I can't do everything. We need support. So microbe.tv/contribute. We are a 501c3 tax-free organization. So in the U.S., your donations are U.S. federal tax deductible. Before we get into our paper today, which Jason is going to tell us about, this is Jason's last episode of TWiN. Oh my gosh, Jason, where are you going?
Yeah, well, last official episode. Hopefully you'll invite me back. But can't believe, 60 episodes. I think it's been 5 years now that we've been going. So, a good stint. But all things have to come to an end at some point, I guess. But we'll be helping out with a new podcast that's going to be supported by the Simons Foundation called The Transmitter. And so stay tuned for that. It'll also be scientists talking to scientists mostly. But I just want to say, Vincent, thanks to you for hosting these podcasts. It's more important than ever that we get the science across to the world. And as you said, this is scientists talking about science. There's no agenda. There's no bias. There's no influence. There's no bottom line. None of us are getting paid to do this, and we do it because, scientists love talking about science, so that's never a chore. But we hope that listeners get something out of this, even the non-scientists. I think hopefully they also get that we love what we do. We do this because we're about discovering new things. And some of the things that we discover can also help in the end. And we never know where that's going to have impact. And the science that we talk about can be very esoteric. It can be, something that just scientists get a kick out of it. But every so often, some of those discoveries can revolutionize medicine, revolutionize technology. Anyway, I hope that TWiN continues as it has been. It's been a fun ride.
Yeah, thanks to Ori Lieberman for starting it. He approached me 5 years ago and said, let's do a neuroscience podcast. He was at Columbia at the time.
Yeah, that's right. And that was, during the pandemic where things were certainly a bit crazy. And I think it was a good distraction for a few of us to think about something that was constructive. And so thanks to Ori for roping me in there. And it's been fun.
So we'll keep an eye on your new podcast, and Jason, hope it does well. Good luck. And we'd love to have you back from time to time, right? So if you see a paper you think would be great and you're not going to do it over there on Simon's podcast, what's it going to be called? Do you know?
Well, that's the transmitter, which is their big neuroscience science communication. I'm not sure there's going to be. I don't know what the name of the podcast is yet.
Transmitter would be great.
Yeah.
Or transmission. Well, yeah. If you see a paper, if not, we'll bug you from time to time. That would be great.
Yeah, absolutely.
So this is Jason's last paper as an official full-time co-host of TWiN. So go ahead. This is really a cool paper, Jason.
Yeah. So this is a recent paper published in Nature by some of my favorite scientists, Paul Franklin and Sheena Jocelyn. They're in Toronto in Canada. And it's on a topic, of course, that's dear to my heart, and that's memory. And we've had a few papers over the years about different aspects of memory. And this one is entitled Systems Consolidation Reorganizes Hippocampal Engram Circuitry. So that mouthful, basically the gist of this of this paper is trying to figure out what happens to our memories when we consolidate them over time. And this is both at the molecular level, the circuitry, the synapses. How do they change when you learn something? And we've known for many years, of course, that, or decades, that the way we encode memories is not exactly how we remember them. There's changes that happen over time, and that's why, in the end, our memories are not very they're not very reliable. We update those memories every time we recall them, but exactly how and why our memories change over time is still not exact, has not been well defined. And so there's a couple of sort of cool things that they bring into this paper, but the one thing that they kind of talk about as a feature, which I would say is maybe a little controversial, which is that the one thing that happens to our memories is that they become less specific. And one idea is that that's because you want to recall memory in a way that's easier to jog your memory. So if you always think about things in the past, sometimes your memories get jogged by a stimulus or something in the environment, or something that someone said that may not have anything to do with the exact memory that you encoded in the beginning. And so that they call this a gist, that our memories become more promiscuous, in that the recall event that triggers that memory may not be as specific as the initial training that you had. And so
so Jason, would an example be you meet someone for a brief time, and then many years later you meet someone else and you think it's that original person? That could be that?
Right. Yeah, exactly. So that's sort of a generalization of that initial.
I'm going to use that as an excuse in the future.
Yeah, you say, well, my brain's built to do this.
Exactly. It's a feature,
and that's why, I confused you with someone else. Or, oftentimes we think about similar experiences that we have, and they sort of merge together over time. And so, some people will remember different aspects of those experiences, and they'll come back to you and say, "Oh, do you remember, when we went and visited the Grand Canyon?" And you were like, "Oh, that was a couple of years ago, right?" And then they're like, "No, that was 10 years ago." So why that actually happens is unclear, and the paper sort of goes into this. And saying that perhaps this is a feature of memory, and that we need to have this.
That's very controversial. I would say that's controversial, because they actually tie it into the reason. They actually propose the reason why you have memory at all, why memory is useful, is so that in the future, when you see a similar situation, you can do some prediction about what would happen.
Yeah.
Right.
But they said that for the prediction to be useful, it's very unlikely for you to encounter exactly the same thing again. So you do want to generalize a little bit so that when you see something similar in the future, you know kind of what to do. But they also notice that this generalization seems to occur more and more as time passes. So like like what I ate for breakfast yesterday, I can probably still remember today. But as time passes, I tend to maybe merge it with some other breakfast I could have had.
Right.
So for me, the whole thing seems like it's forgetting, but they're saying it is not the same as forgetting. It is actually generalization, and it is a feature, not a bug, which is very interesting kind of way of looking at it.
Right. And this group, I mean, the Franklin group especially has been working on forgetting. And, we often, so forgetting can be many things. One, it could be that we can't retrieve the memory is there, but we can't access it. It could be that the encoding degrades the initial encoding event, that information is degraded so that it's gone. Or a sort of unexpected aspect of memory is that forgetting is an active process. So it's not that it's just degradation of the system. It's that there's an active process going on that makes us forget, and if that's the case, then it's a feature. It's not a bug or an outcome or an inevitable outcome. And that's something that the Franklin Labs proposed many times. And actually, they had a really interesting paper a few years ago talking about why we can't remember anything that we experienced as a baby, or even as an early on and as a kid. And they had proposed a sort of hypothesis there that it's because when your brain is reorganizing and they have this massive amount of neurogenesis and synaptogenesis in the beginning, that you need this highly plastic circuit to sort of allow our development to occur, and that gets in the way of consolidation. And there's a similar theme here, as we'll get to, about neurogenesis, and that's also controversial for adult mammalian brains. So, of course, they're talking about experiments done in mice. This is not in humans. And there's a couple of themes here that we've talked about before. So the main behavioral paradigm they're using is something called contextual fear conditioning, where essentially you put a mouse in a box and they give them a shock. And in the box, there's some cues like visual cues, olfactory cues. So if you put them in the box, an hour, a day later, a couple of days later, they freeze because that's their fear response. And it's a direct output of them, we think, remembering that they got shocked in that box. And you can get a little fancier now. So they have a box that you get shocked in. That's the box A. But on the recall test, they can also put you in a box B, a different box that has different cues. That's a box that they've never been shocked in. And if the memory is specific to the box that they got shocked in, then they shouldn't freeze in box B. And so they do this discrimination task. So they basically allow you to sort of look at the specificity of the memory by putting the mouse in these two different boxes. So as you'd expect, a day later after the training, there's a big difference in the amount of freezing that you see in box A versus box B. But then they look 2 weeks later, so 2 weeks after the encoding event, and there's still a significant difference between box A and box B in terms of freezing. But then on what they're calling remote memory or remote recall, so 28 days, a month later, what they see is that there's now no difference between box A and box B. But not only that, they freeze a lot. So it's almost like you put them in box B and they're kind of like, maybe I got shocked in this. I'm not sure, but I'm just going to freeze anyway.
Yeah. I think it's really nice. This is a really nice paradigm because you can look at two different things. The first one is earlier we were talking about whether the mouse forget versus generalizing, and you can ask, did the mouse forget that it used to get shocked at box A? And you can just look at did the freezing decrease in box A after a month? And in this particular experiment, it didn't. The mouse was still freezing to box A, but what happened was that the freezing to box B dramatically increased from 1 day after training to a month after training. And that's how you can say the mouse actually generalized from A to B, because the B freezing in B went up a lot. So it's a very nice paradigm.
Exactly. So that's the main sort of phenotype or behavior that they're going to test throughout the paper. The next sort of tools that they use are also probably familiar to listeners, and this is the engram technology. So essentially, this is tagging the neurons that are active during that training period. So we of course think that the active neurons that are sort of processing the experience, or are the neurons that are ultimately incorporated into the memory circuit, and that's called the engram. And that's this technology has been now used in many different labs. This is using this immediate early gene called FOS, which is induced by neuronal activity. They use the promoter. There's a mouse line that now you can use to drive whatever expression of things in those active neurons. So in the first experiment, they just looked at which cells are activated during that encoding. So they tagged them, and then they wanted to look at in the recall event, the amount of overlap in the active neurons in that recall event with the initially encoded active neurons. So the idea is that you have, let's say, 10 neurons that are active during the encoding event, but on the recall event, there's going to be a subset of those cells that are also active. And we think that subset is the engram, in that you need those cells to be activated on that recall to get the memory recalled and the behavior output. And they do that for both...
assuming the memory is stable.
Assuming the memory is stable. So, there's been previous
There's controversy with that too.
Yeah, I mean, there's definitely been lots of experiments that show even at the 28-day time point, you can reactivate or recall the memory if you just activate those hippocampal neurons that were tagged in the beginning.
That's true. But then I'm going to channel Vivian, because although Vivian can't be here, she sent in some questions. One of the question was, in one of the earlier TWiN that we did, I think, Jason, you presented this, that showed in the olfactory cortex, they tracked the set of neurons that were active when the mouse was smelling a particular smell, and the set of neurons drifted over time.
Right.
So I think in the cortex, there's some drifting that people can't quite, we don't know what to think about this in terms of why the engram itself can move a little bit.
Yeah.
But it sounds like in hippocampus, at least for the contextual fear conditioning, it perhaps seems to be stable.
Yeah, and this is something that we're interested in my own lab, is sort of the stability of memory at the molecular level, and sort of this conundrum where we can clearly tag these neurons and they seem to be sufficient to induce this recall event even a month later. Yet despite the fact that when you do measure the neuronal activity from these circuits, from these cells, the activity changes, the pattern changes over time, and as you say, it's called this drift. I mean, and the other thing that is sort of still a little bit controversial is this idea that they bring in systems consolidation. The classic view of systems consolidation was that the hippocampus is important for making the new memory initially, but that eventually the storage of that information goes to the cortex. And the long-term storage is in the cortex, because if you ablate the hippocampus, or even in humans where you have Alzheimer's disease or some temporal lobe dysfunction that wipes out the hippocampus, you can still recall those really old memories. But I think one of the sort of newer ideas about this systems consolidation idea is that, okay, maybe the cortex is important ultimately for the storage, but that you still need the hippocampus to specifically retrieve or make the memories make sense to you in a way that's important. And that's kind of what they get at here, which is that even at this remote time point, there's active influence of the hippocampus on the memory recall events, which we'll get into. So this overlap. So basically what they just show in this first experiment is that, as you'd expect, you get more overlap with the initially tagged neurons in box A because, that's where they got shocked. But as the time goes on in the remote memory stage, there's now no difference in the amount of overlap when you put them in box A or box B. So again, suggesting now that the engram, the circuit, is now more promiscuous in that the amount of reactivation or recall is consistent with the behavior now that you get the same amount of box B as box A. So that, so they're like, okay, well, there's some sort of functional reason for the behavior. The engram overlap is now, no different between A and B. And so maybe it's the engram itself that's causing this generalization because the same amount of cells are reactivated in box B as box C. And they kind of then want to know what's happening at the neuronal activity level, at the structural level. They do some cool experiments now where they add a GCAMP, a calcium indicator in the CA3 axon. So just a reminder that the hippocampus has this very stereotyped structure. The input comes into the dentate gyrus, and then the dentate gyrus sends information to the CA3, and then CA3 to CA1. So it's these three major synaptic targets, and they want to know where in the circuit is this generalization happening. It's been thought for a long time that the dentate gyrus is important for this discrimination aspect or making the memory specific. But they were sort of agnostic to where the changes were happening in this case. And so they were able to record from these inputs coming in from the CA3 to the CA1. And they kind of look at this correlation with behavior. So they can sort of look at the freezing bouts in the box along with the neuronal activity. And they notice that, as you would expect, that there's more activity that correlates with the freezing bouts in box A initially than box B. But at the remote time points, they see these events that correlate with the freezing in both box A and box B. So this is all consistent with that the engram circuit changes over time and it becomes more promiscuous. There's more activity in box B than in the recent memory recall events. And then they show that so the nice thing, of course, now in neuroscience is that we don't just have to observe activity. We can now specifically manipulate it with optogenetics, these channels that are light sensitive. So they put some light sensitive channels in that CA3 input to the CA1, and they block that activity. And when they do that, what they see is that now there, as you'd expect, there's less freezing in box A and in box B. That experiment that's not unexpected. Of course, we know that these inputs are important for the recall event, or but what they see is that you basically can inhibit both this generalization, this amount of freezing, in both boxes. So the CA3 to CA1 input seems to be important for the promiscuous or generalization of the memory, which again is not unexpected. So, but that sort of proof of concept that they can do these kinds of experiments.
Jason, do you have any hypothesis about, instead of inhibiting these engrams to show that you reduce freezing, if you were to activate these engrams, which I think other labs have done before, if you activate these engrams in a different context, you can cause the mouse to freeze because it kind of thinks that it's in the old trained box. Is it because the mouse literally sees that it is in the new box, or does that just increase like a general fear that is somehow learned? Like how if I want to gain some sort of intuition, is it?
We don't know. We can't ask the mouse what it's exactly experiencing. But what I guess what we think is happening is that the mouse is experiencing as an event as if it's seeing the cues that it would associate with the shock. Okay. So we think it's specific. It's not just this general anxiety effect, because if you do this in non-engram neurons or random neurons, then they don't freeze. So it's not just that you're activating, enough cells. There is some specificity built into it. I will say that that initial encoding event activates about 10 to 20% of all the cells in the hippocampus. So you're capturing, but that's not the final engram. The final engram is probably much, much smaller, as you can see with the overlap percentage that they're getting. It's at most 10 to 15%. So then they go, okay, well, what's happening at the synaptic level? And so this is this is quite cool, this next set of experiments, because most engram papers, they look at the cells that are activated, but they're not looking at the specific synapses. But here they use this another tool called GRASP, which essentially is that they have a split GFP, so the fluorescent protein is split up, and it's not fluorescent unless the two parts of the GFP are bound together very closely. And so what they do is they express one part in the presynaptic terminal and the postsynaptic terminal. So there's two different sides. And then they still take advantage of that activity promoter to tag the cells. So basically they only get fluorescence in cells that are active, and when they're connected to a postsynaptic partner. So if they see GFP fluorescence, it means that synapse was probably activated, or it's part of the engram tagged cell.
I just want to quickly highlight GRASP as being maybe the best acronym I've ever seen. It stands for Green Fluorescent Protein Reconstitution Across Synaptic Partners, which spells basically GRASP. And for it to work, and literally as Jason described, your presynaptic terminal express half of GFP protein, your postsynaptic terminals express the other half of the GFP protein, and they have to be in really close proximity, almost shaking hands, for the green to be able to kind of glow. So you're kind of grasping each other as a partner. So yeah, really good acronym. And
the thing is, and I will say, like, this technique has been around for quite a long time, but it's hard to get to work. So this lab did not develop the technique. So it's great to see that another group can get this technique to work, because I think it's. Now, they actually take advantage of one of the things that makes this technique not great, is that when you get the bound partners, it's irreversible. So, basically the idea is that you're looking at the synapses that are on these engram neurons, they don't. So whatever changes you see over time are due to other factors, like maybe those synapses are pruned, maybe those synapses are lost in some way, and so they take advantage of this irreversible aspect of it to look at how do the synapses change over time in those engram neurons.
Sorry, Jason, just quick clarification. When you say reversible, you mean once the GRASP protein has been starting to express in the engram neurons, they're going to continually getting expressed. Not that once the GRASP has formed across a synapse, that synapse is permanently undeletable. Because I can imagine
the latter. So once you get the two sides connected to each other and you see fluorescence, that interaction is permanent unless the whole synapse is removed.
Oh, even when the synapse is removed, I see what you mean. Oh, that makes it tricky.
Once the synapse is removed, of course, then the proteins are degraded. But the idea is that fluorescence, once it's in that state, it'll last. So one could have said, oh well, we see these synapses formed in training, but maybe nothing's stable. And as you sort of suggest, by 28 days, maybe we don't see any synapses. But that's not the case. So this is one of the first sort of examples of using this kind of technology to say, well, there are some synapses that we tagged during training that are still there 28 days later. So that in itself is actually a fairly big finding. And so they also now look at different synapses. So the first set is the dentate to the CA3, and because they want to know where in the circuit are these synapses changing over time. And so the dentate to CA3, they actually don't see any differences between the number of synapses that are observed in the recent versus remote memory recall event. And when they look at the CA3 now to CA1, now they actually see a difference where they see more, unexpectedly, they see more synapses in the remote time point than the recent. And that to me, that's unexpected. I would not have predicted that. But that goes along now. They're saying, okay, well, there's more synapses that we can see that are there at the remote time point, maybe because there were more synapses that were formed during that consolidation period. And consistent with this idea that these are involved in some sort of memory process, the synapses are not clustered randomly. They actually they're clustered quite close together, which is another hypothesis about how memories are formed, that you have specific clusters of synapses. Here the interesting thing is that they see more of these clusters, or the synapses that they tagged become more clustered over time. So that's also unexpected and kind of interesting.
But I think they found in figure 1, so we're now on figure 2, but in the previous figure, they found when they tried to look for the overlap between the engram cells during encoding and the retrieval cells at the remote 28 days later time point, they found that the promiscuity, like the context B, the second context, it activated more cells in CA3 and CA1. So those two connections already started between those two, like the connectivity between CA1 and CA3 and CA1 seems to already been boosted.
So maybe I guess I neglected to say that what they mostly looked at in the synapse level here is that they couldn't obviously look at synapses that were specific to either A or B. They could only mark A or B when they did the recall event, and so they're only labeling here the synapses they see in the context B. So what they see is that this increase in synapses, or the difference between remote and recent, they see in this context B, which is again consistent with this idea that there's somehow some more more promiscuous or generalized engram formation over time, or restructuring of the engrams. I can't remember if they have in the supplemental data the context A, because that would obviously be useful to know if they is there a difference in the remote memories.
Yeah, I don't think they did look at context A ever in terms of the synapses. Yeah, that would be interesting.
Yeah, which is kind of surprising. I would have asked that as a review. It'd be
maybe other people have done it before.
Maybe
although with grass, it might be tricky.
Yeah. I mean, I know these experiments are very difficult. Okay, so it's all sort of lining up with this idea that to get this more generalizable memory or just memory at the remote time point, there's restructuring of the engram, and there seems to be perhaps more activity that's related to box B, and where is that coming from? So what is the sort of driver of that change? And based on their previous work, as I mentioned earlier about neurogenesis, they hypothesized that maybe these synapses or input that is coming in over time is driven by new neurons that are born in the hippocampus. And as I said, this is sort of a controversial finding, or a general idea that you, as an adult brain, you have new neurons that are formed. This absolutely happens in rodents. There's obviously lots of data for this, but whether it happens or how much it happens in humans, there's been conflicting findings. Of course, we can't do these kinds of very detailed molecular manipulations in a human, so it's mostly due to, can we observe new neurons being born in adult tissue? And that's not a straightforward, thing. And so the most recent sort of findings have been using the RNA sequencing and different ways to identify newly born neurons. And those new techniques, basically, some people find no new neurons in adult brains and others do. So jury's a little bit out of how important this is, but what they
we never generalize in humans.
What they claim is that this is important in this context for mice. And so the way they do that is they use the tagging approach, the grasp approach, and then on top of that, they add another virus that has been routinely used to label newly born neurons in the dentate gyrus. And then they want to know, and so they also, many labs have shown in the mouse dentate gyrus that these newly born neurons can get incorporated into the engram circuit or new circuit. But this process takes 4 to 6 weeks.
Jason, can I quickly ask a question? So like these newborn hippocampal neurons, do they largely go to the dentate gyrus and then send projection to CA3, and they don't go to the CA3 and send projection to CA1?
Exactly. So almost all of them are only in the dentate. They don't move to...
Do you know if dentate gyrus has a higher death rate in neurons compared to CA3 and CA1, so that there's kind of balance in turnover? Or is my dentate getting bigger and bigger, at least in a mouse?
Yeah, so that's a good question. I would say, one would say the amount of neurons that are being made is still small, fairly low. Yeah. Okay. But that's another sort of the size of the hippocampus can change over time, or even with experience, or even
if
in
you're
humans,
taxi
just
driver,
documented changes. There's like this classic paper that decades ago about taxi drivers in London that have bigger hippocampi.
Not anymore with Uber and with GPS.
Right.
Yeah.
now they want to know, can these newly formed neurons or newly born neurons form contact synapses with the engram neurons in CA3? And indeed they can. And not only that, but then now they can look at whether the synapses that change over time are influenced by neurogenesis. And the way they do this is that they radiate the mouse. So again, this is sort of a very crude way of blocking dividing cells. They give the mice a big dose of radiation, which has, of course, its own caveats. And there's, some controversy over obviously side effects, or not controversy, but there are caveats with side effects of irradiating a mouse. But this stops this dividing newborn neurons from happening. So that's definitely the case. And then they look at ablating neurogenesis. Can it affect the behavior of the mouse? And whether they can also see a change in this overlap. So the first thing they look at is the engram overlap. So as just a reminder, the recent overlap, of course, for A versus B, is that there's much more activation of A than B, but in the remote memories, A and B are equal. But what they see here is that if they ablate neurogenesis during the training period, now A and B are much different, just like in the recent time point. And they maybe attribute this to interneurons. So as you mentioned, the dentate, these newly born neurons don't necessarily go all the way to the CA1, but they can also target interneurons that may influence neuronal activity in the circuit. But they also don't see this change in synapses in the CA1 with when they ablate the neurogenesis. So the distribution of synapses seems to be the same in presumably as A, although I don't think they ever show, of course, that what the A specific engram synapses look like. So given that the radiation has issues, they then wanted to say, well, okay, can we promote neurogenesis? And if we promote neurogenesis, what happens? And a classic way to promote neurogenesis is exercise. And
that's why humans don't have neurogenesis.
That's true. Maybe.
That and also stress also reduce neurogenesis.
But so humans exercise, right?
Right. Not enough.
So again, of course, exercise is a very non-specific manipulation, but certainly one thing that it does do is increase the number of cells that are born, at least in a mouse. And to induce exercise in a mouse, they just put a wheel in the treadmill, essentially in the box. And the mouse loves running on it. It'll run miles and miles.
They run even in space. Have you seen videos of when they put mice up on the ISS, International Space Station? No. And the mice are like, they don't even have a running wheel. They just zoom around like the perimeter of the cage. Awesome. It's amazing. Yeah.
Wow. Okay, so one would predict that perhaps if you increase neurogenesis, then maybe you would accelerate this change in synapses or structure of the engrams. So now they looked at that intermediate time point, the 2-week time point, rather than the 28-day time point, thinking that maybe then they'll see something similar to the remote time point when you increase.
Just a quick reminder: during intermediate time point, based on previous experiments, they showed that the mice did not generalize yet in this time point.
And the overlap between A and B was different still. So there was more specificity for the overlap in A versus B. Mm-hmm. But here what they see is that, at least in the first experiment with the engram, is that the overlap now looks similar. There's no difference between A and B at the intermediate time point. And And in the synaptic alterations, they see a similar redistribution of synapses that looks like that remote time point in context B. So accelerating neurogenesis accelerates this restructuring of the engram. And then the final figure here in 4 is what happens to the behavior. So now they do the same experiment. In the first experiment, they irradiate the mice. They look at A and B and the different time points. And so now, without neurogenesis, the remote time point, there is still they don't generalize. There's specificity built in where they only freeze to A and not B. And on the flip side, when they increase neurogenesis with, again, running, which again has different, caveats, that they see this generalization occur much earlier in that intermediate time point. So now there's no difference between A and B in terms of freezing, and it's similar to what you see in the remote time point. So this is surprising to me, but in a way that, again, comes back to that question of is this generalization a feature or not, or is it a bug? And so this is an active process. They're saying that the default state, if you don't have neurogenesis, is that the memory remains specific. Now, if I had, so lots of lots of labs use the same contextual fear conditioning to say that this discrimination is accelerated in aging mice or disease-related mice, where they're saying, well, you lose the specificity of the memory very quickly, and that's a bad thing. So in this case, what they're saying is that maybe this generalization is a built-in aspect of memory consolidation. Which is provocative again, and sort of saying that this is something that the brain wants to do. It's not that like you start to see disease aspects or generalization.
But in the disease mechanism, is it because the mice forgets faster? Like, does it freeze less in context A, or is it generalizing to context B? And if it's the latter, presumably it's via a mechanism different from neurogenesis, right? Because I definitely think there's been. I mean, you see both, right? So sometimes there's no freezing because they don't recall anything, or they do see this accelerated generalization where they freeze to everything. I think the drawback. So this particular paradigm, this drawback of this paradigm is that it's a very simple memory. It's associative memory. Mice that are deprived in these environments, when you get shocked, it's like the biggest thing that's ever happened to you. And so the question of sort of how does this relate to other kinds of memory? So they do look at spatial memory in some aspects of this that are, where they look at this, what's called a Morris water maze, where I think we may have talked about this before, but the one way to test spatial memory in a mouse is that you put them in a pool, and they hate swimming, but the pool is opaque, and so then they swim around, and then they find a platform that's underneath the. You can't see it, but when they land on that platform, of course, they're happy because now they don't have to swim. And over time, they can remember where that platform is. So every time you put the mouse back in the pool, they'll swim straight to that platform by looking at the visual cues around the
using visual cues, exactly. So they had looked at that kind of memory paradigm and see something similar in that there's more generalization happening in the two different kinds of boxes or pools with the different cues.
But I like the context thing better, because in water maze, you can't tell if the mouse is floundering around where the platform used to be. Is it because it forgot, or is it generalizing?
And there's also one of the problems with the pool is that they're stressed because they hate the soil.
But they're also stressed here. I think the authors they discuss in the discussion that they really are just looking for both these experiments. For this paper, they're looking at a very kind of aversive memory. So they didn't claim that it generalizes to everything.
I do this experiment in my backyard. I have a pool, and I always found dead mice in it, right? Because they can't get out. But I put in these 2 platforms, and now no more dead mice. So I wonder if the same mice fall in and they get their cues and they know where the platform is and they know where to get to it.
But what do they do? Do they go to the platform and then jump out from the platform?
No, you can just crawl up. There's a little ramp.
Oh, wow. That's very nice of you too.
Yeah, there's a little floating thing that they can easily crawl up on, and then there's a ramp. And the same with snakes. I mean, I used to find snakes all the time, and they apparently go up. I feel better. I feel
snakes and ladders is real.
Yeah, but the cues, there are plenty of cues, obviously. So if they fall in again,
that's very nice. You turned your pool from a death trap to like a bathtub for mice.
Yeah, I don't want things dying in it. It's very sad, right?
Yeah.
Yeah, well, so that's pretty much the experiments. I mean, there's some details that we glossed over, but that's the main experiments. The final experiment they did is just to show that this neuronal activity, this restructuring of the circuit, is also influenced by manipulating neurogenesis, consistent with this idea that perhaps one of the drivers of this restructuring over time is these new neurons being incorporated into the circuit.
I just realized why it's called gist memory. You get the gist of it.
Right.
Yeah, you don't remember the whole thing. You just remember kind of the main dimensionally reduced 2-dimension of PCA, whatever.
So Jason, we were talking earlier about, we talk to scientists here, but how could you summarize this in 1 to 2 minutes for a layperson? Because then we're gonna make a TikTok out of what you say.
Yeah, I guess TikToks are the way to get the young folks listening, right? Yeah, I guess the one way to summarize this is to say that we think that our older memories become more less precise over time, and that's because we think that that's a form of forgetting, or just an aspect of memory that's unavoidable. But actually, what this group found is that the default is that the memories are quite precise, but that there's this added complexity over time where you have this generalization occur. And they're saying that this is a feature of memory, and that we want to be able to reactivate our memories in a less precise way over time, because that helps us predict the world, and you don't want those memories to be so hard to recall. Now, that all of that's hand waving, but what they do show is that in adult brains, there's part of the structure that's required for making new memories, where there's newborn neurons that can influence how those memories are stored and recalled. And that's a big surprise. Now, this is all done in mice, and we don't know if this is happening in humans. But we certainly know from psychology experiments that our memories are definitely less precise over time. But the most important memories are the ones that we can recall for up to a lifetime. And so the brain is constantly trying to balance the specificity of things with plasticity, or being able to learn new things. And so whether this neurogenesis, these new neurons that are contributing to the memory, is occurring in humans, or if there's some other way of influencing memories, that still needs to be looked at.
So it's a feature, not a bug.
It's a feature. Losing our having just memories, the GIST kind of memories, is perhaps a feature.
So Jason, what happens to memory in Alzheimer's?
So in Alzheimer's, really the first thing to happen is that you can't make new memories. It becomes very difficult to make the memory. And so and especially this conversion from short-term to long-term. So some of the first symptoms that happen is that, you forget where you parked your car, or you forget a phone number very quickly, or forget a name that someone gave to you. But over time, making those new memories becomes harder and harder, but they can still recall the old memories that they have. So it's kind of interesting in that there's a time dependency. As the disease progresses, it's the most recent memories that get degraded. And so by the time that you're at the sort of end stages of the disease, the only memories that you can recall are the very old ones, the most important ones, perhaps like childhood memories, or if you've been married for a long time, you can remember getting married, but the more recent ones, like even if you've had kids, you start to lose the ability to recall your kids, and that sort of time dependency of memory degradation has led to this idea that the storage of those old memories happens in a different place. It's not in the hippocampus, because we know in Alzheimer's the first target, the first disease part of the brain is the hippocampus, and that's why you can't make those new memories. And something that this paper does not really go into is that they've only looked at this restructuring in the hippocampus. We don't know, how that affects the cortex, which is where we think ultimately the really long-lived memories are stored.
So in this paper, they used the 28-day point where the memories become less precise. Do we have any sense for how long it is in humans?
I think it's something similar. I think the time point of, about a month is when you start to really have either you completely forget that memory, or it is stored in a long-term storage. The one thing that this month period is, I think, it also coincides with this neurogenesis idea. So these newly born neurons take about a month to get incorporated into the circuit. And what happens after that, though, it's not, then it's sort of unclear.
So Vivian had a good question here. I'm thinking about people who have photographic memory, assuming this is the same as precision-heavy memories, or at the extreme, individuals with autism who remember everything. Could we hazard a guess as to how their predictive abilities are?
Yeah. So that's a really interesting. Yeah, that's a really interesting question. So we know that there's a subset of humans even that have this remarkable ability to remember anything that for up to a year. Yet they don't seem to. In some normal cases, we're not talking about the autism cases. They seem to function normally in society. It's not like if you have this super memory that you're stuck. Because one idea was that if you recalled every single thing that happens to you, you just get lost in all that information. Every time you recall something, you're like, oh, I can. So the brain, obviously, when we're going through life, it's not like every second we're getting bombarded with recall events. So there's probably something there that suppresses unless you have to recall the memory, you're okay. Now, in the case of autism, that's where I think some of the savant-like people, they have this very unique ability to do math, or they can recall numbers, or they can recall abstract things, but at the expense of other things that the brain normally does, like social interaction or empathy or like emotional valence. That's all sort of gone. Now, we don't know if that savant-like activity is because of losing those other abilities, or is it just that the brain is functioning in a different way.
Amazing.
I wonder. Sorry, quick question. I wonder, since neurogenesis seems to be regulated in mice by environmental factors, like, exercise, like we talked about, but also like stress, or like people have found stress and depression, I think both reduce neurogenesis. Whereas things like environmental enrichment, like if you live in a nice house with a lot of stimulation, you get a lot more neurogenesis. Does that actually, if the sole purpose of neurogenesis is to allow us to generalize our memory, does that make sense that it is regulated by these things? I wonder.
Yeah, well, I will say that, I mean, people always ask me, what should I be doing to keep my brain, intact as I age? And regardless of whether neurogenesis happens in humans, exercise is probably the best thing to do for your brain. Well, generally for your health, but also for your brain. But there is also a fair amount of evidence that you need to keep your brain active, right? So one thing that happens as you get old is that you lose some of your friendship groups. You go eventually into a retirement home, and your loneliness or being separated from society seems to accelerate that aging process of the brain. And actually, there's some of the interesting anecdote that I have here, or not, that I learned recently. There's some societies where it's not like they stick their older people alone. They live with the family. They live, you have Asian societies or Pacific Islanders where you have generations of people in the same house. And the markers of dementia are actually quite less in those populations. Now, it's not clear. So there's one. It could be that this is because they're staving off dementia, or it could just be that the dementia symptoms are masked by the fact that they have this big support network at home. But I think there could be something there where keeping your social groups intact, being able to have complex interactions with people keeps your brain activity high, and that's good for lots of things, and maybe it's also good for keeping those new neurons being born. But I think this idea, they are saying in this paper that this feature is good for predicting the future. Now that's probably quite a far stretch.
Yeah, I think that's theoretical. Yeah, because in fact, if you need to discriminate box A from box B, then this is actually going to be worse for you.
Yeah. I mean, I guess the sort of in a normal environmental setting, where let's not talk about humans, but let's say before we had technology, the dangerous animals or coming into dangerous settings, you probably want to trigger those more than not.
That's true. That's true.
It's better for survival if you freeze, even if you think maybe this was where you found a lion or whatever.
Well, that's why I'm going to podcast forever, so I can stave off this dementia, because I'm thinking all the time about different things.
Yeah, no, I mean, exactly. And I think doing new things is also important as you age. Go to new places, travel. If you've never done a specific kind of word puzzle or word game or something like that.
Ah, okay. I'll travel more, Jason. Word puzzles are something I never did, but maybe I should try them.
Yeah. I mean, they engage your brain in a different way, and it's useful.
Cool. Right. Thank you so much. That was a great paper, Jason. Good way to end.
Yeah.
TWiN.
Well, again, I don't want to, sign off without saying thank you. And it's been great. And Tim and Vivian, it's been great to have you two involved the last few years, and oftentimes jogging my memory about, oh yeah, we have TWiN today, or who's doing papers. So, it's been great to have you all.
It's TWiN number 60. You can find the show notes at microbe.tv/TWiN. You can send any questions or comments to twin@microbe.tv. And please consider supporting us so we can continue these science programs. Microbe.tv/contribute. Jason Shepard is at the University of Utah. Jason Synaptic on X. Thank you, Jason.
Thanks, Vincent.
Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent, and thanks, Jason, for all your work on TWiN and all the papers you presented. I definitely learned a lot, even though they all kind of joined together into a giant gist of things. But yeah, definitely learned a lot. Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.